Now we'll do the remaining part of connective tissue, which is blood. Blood is what is called an atypical type of connective tissue. By definition, connective tissue has to have ground substance, it has to have fibers, and it has to have cells. In blood, you have ground substance, which is plasma. You have cells, which are the different types of cells that you see, the red blood cells, white blood cells, and platelets. You have fibers, which are called fibrin threads, but fibrin is not seen normally. Fibrin is only seen when blood clots. So clotting of blood releases fibrin threads. Since it's not seen all the time, that's why we call blood an atypical type of connective tissue. Okay? So the ground substance is plasma, which forms 55% of the blood. You'll do a little bit more about blood when you do A and P too. For now, that's all you need to know. Fibrin are threads which are seen when blood clots, and they help to kind of form the clot. The various cells that you see, and these are important, and as always, look at it. You have to know features, you must know functions, you must even know numbers wherever given. Red blood cells. Red blood cells are also called erythrocytes. Their main function is to carry oxygen, and that's because they have a pigment called hemoglobin. So they have this hemoglobin pigment in them, and that hemoglobin has an iron component which is able to attach to oxygen. So that's why red blood cells are able to carry oxygen to the tissues. Red blood cells are the most plentiful of the blood cells. They are in millions. The next group are white blood cells. White blood cells are collectively known as leukocytes. So white blood cells are collectively called leukocytes. And they are divided into two groups. One group with granules, so these are known as granulocytes. Another group without granules, which are called agranulocytes. The granules are present in the cytoplasm. So granulocytes have granules in the cytoplasm. Agranulocytes do not have granules in the cytoplasm. So let's take a look at the picture. So up here in both these, you can see these are red blood cells. All of these are red blood cells. These are beige to light salmon in color. Notice they do not have a nucleus, red blood cells. The central region is pale, because red blood cells are biconcave. So if you look at a red blood cell from the side, it's like biconcave. So the central area is much thinner than the periphery. So that's what makes this area pale in the center. And see how many red blood cells there are, because they're in millions. White blood cells are in thousands. About 11,000 is the higher range. And let's look at these cells. So notice this cell, you can't see the granules that well, maybe not from the distance, or you can. So you can look at this part here where the granules are colorless or neutral. Look at this one where these granules in the cytoplasm are red or pinkish. Here they are deep pink, but they could be reddish, orange. And then here, look at this third type of cell where, again, you look at the granules. Notice these granules are purple or blue. So in these three cells, you can see that the cytoplasm contains granules, so we call them granulocytes. These two cells, notice the cytoplasm. No granules. Look at this one. No granules. So these are agranulocytes. So let's look at the cells which belong to granulocytes. They have the word phils, P-H-I-L-S, phils at the end. So the first of these granulocytes is something known as neutrophil. So here is a description, or in this image you can see a neutrophil. Here's a neutrophil, Here is another neutrophil up here. Here's another neutrophil. So in a neutrophil, the nucleus, which is this dark purple structure, instead of being single, just like one blob, notice that the neutrophil seems to be broken up, and this is known as lobes. So the neutrophil has many lobes, anything from 2 to many, to 5. And the granules which are present are colorless, or when they take up the histological stain, they look neutral in color. You can't really tell the color. So that's why these are called neutrophils, neutral colored granules. They have a lot of cytoplasm, which you can see here. So whenever a cell has a lot of cytoplasm, it is phagocytic. Remember, phagocytosis was a form of active transport using ATP. So these are phagocytic. So here's the function of neutrophils, phagocytic. Typically takes part in bacterial infections. It usually phagocytoses bacteria. Second type of blood cell is an eosinophil. It's called an eosinophil because its granules take up a pink stain, maybe light to really dark pink, or maybe even red, or anywhere from orange to red to pink. Stain could be anywhere like that. That's because there is a histological stain called eosin, which is pink in color. So this eosinophilic granules look like that, and when we stain it with something else, not with eosin, but because it looks like that, if it was stained with eosin, it would take a pink color. This is why this is called an eosinophil. So pinkish granules, but could be red, could be orange red, so it can vary. Its nucleus usually, or rather typically, is described as being bilobed. So two lobes, one lobe and two. So this kind of gives it like a sunglass appearance. But it's not necessary that it has to be bilobed. It could be multi-lobed like a neutrophil. The way you'll tell the difference between them is because of the granule color. Okay, so an eosinophil could have a bilobed nucleus but could be multi-lobed. This is important because it takes part in parasitic infestations and allergies. So whenever someone has severe allergies, spring is coming up, pollen in the air, if it's a lot, some of you may suffer from allergies. If you take your eosinophil count at that time, it will be up. If someone has parasitic infections like worms in their body, again, the eosinophil count goes up because it helps to deal with this. Okay, so that's the function. So here's an example of a question. A person is allergic to pet dander. Which cell count would you see increased in them? And I give you choices of red blood cells, neutrophils, and eosinophils. Your obvious choice should be eosinophils. See that? So that's how you kind of put the information together. The third kind of cell is called a basophil because the granules take up a basic stain which is bluish in color, bluish purplish. The nucleus is often hidden because the granules kind of sit on top of the nucleus. These are very few basophils. If you count 100 cells, you might get one basophil. These granules in basophils secrete histamine and heparin. So they secrete a substance called histamine and heparin, which takes part in allergies and inflammation. They are very similar to mast cells because mast cells also secrete histamine and heparin, yes. And mast cells also take part in inflammatory reactions. However, if someone has an allergy, because as I said, basophils are very few in number, eosinophils are the first ones to arrive on the spot and take care of that allergy, and theirs is the count that goes up more than basophils do. Basophils are very, very few. As I said, 100 cells, you're lucky if you find one basophil. But when they are present, their function is to also help in allergies and inflammation. So these were all the granulocytes. Next, we look at the agranulocytes. There the cytoplasm doesn't have granule. It's usually a grayish blue color like this. Doesn't show up grayish blue like over here, but it is usually gray blue like this. There are two cells in this. One is called the monocyte up here, M. This is the largest of the white blood cells. Notice how much cytoplasm it has got. So this monocyte is also phagocytic. In fact, monocytes change into macrophages, which are phagocytic cells. So they turn into macrophages and then they go and phagocytose any cell debris, anything that is around. Their nucleus is often described as kidney-shaped, but it need not necessarily be kidney-shaped. It can be any shape. It can look like a flower, it can look like a horseshoe, could look kidney-shaped, anything, but a lot of cytoplasm. No granules. Second agranulocyte is what's called a lymphocyte, which is this one, with a very prominent round nucleus. Very little cytoplasm. So notice just a rim of cytoplasm. Sometimes you may not even see the cytoplasm clearly. And this takes part in the immune response. It produces antibodies and helps with immunity. So these are lymphocytes. And lastly, we have cells. These are not typical cells in the sense that they don't have cytoplasm and a nucleus which can be observed really well. They're like almost like cell inclusions, but we call them cells. Very, very tiny structures. These are called platelets, so you can see them distributed up here. So these are these tiny structures which you see. They help with clotting of blood. So platelets are very important for clotting of blood. So that was connective tissue. We next move on to muscle tissue. And again, first is classification. So classification based on structure. We call it striated or non-striated. Striated means it has striations which are known as stripes. So if you take a muscle cell like this, when it has this banding pattern, which is called striations, then we call it striated muscle. When the muscle does not have this banding pattern, there's nothing, no lines in it, then we call it non-striated. Then based on function, we divide it into voluntary if it is under your control, and involuntary when it is not under your control. So this is functional classification. Now muscle cells tend to be elongated. They also have the capability of contracting, so they can shorten and get back to their original shape. And a muscle cell is called a muscle fiber. So remember, we did fibers in connective tissue where you saw collagen, elastic, reticular fibers. They were like just thin threads. So do not confuse those with a muscle fiber. A muscle fiber is nothing but another name for a muscle cell. Okay, so it will have a nucleus, it will have cytoplasm, so it will be like any other cell. Only thing is we call it a fiber because these tend to be elongated. And inside a muscle fiber, you have tinier filaments present, tinier structures present, which are known as myofibrils. So notice the use of the word myo with muscle. So inside a muscle fiber, you'll have structures present which are known as myofibrils. And when we do the muscular system, we'll do these in more detail. We also use the word sarco with muscle. Apart from myo, we also use the word sarco with muscle. So the outer covering, the cell membrane of a muscle cell or a muscle fiber is known as sarcolemma. So this is the cell membrane or the plasma membrane. So this is the plasma membrane of a muscle fiber or muscle cell. It's called sarcolemma. The cytoplasm is called sarcoplasm. Muscle also contains a pigment which helps it to attach to oxygen. That pigment present in muscle is known as myoglobin. So red muscle has more myoglobin in it. It gives it a red color. Like red blood cells have hemoglobin, and that's what gives red blood cells the red color. Similarly, myoglobin is the muscle pigment present, and it's seen in red muscle. And because they're capable of attaching to oxygen, they tend to not get fatigued as quickly as other muscles which don't have as much myoglobin. Again, we'll do this more in detail when we do the muscular system. All muscles are supplied by a nerve, and nerve supply is extremely important for a muscle. If a muscle loses its nerve supply, it undergoes something called atrophy, where it dies. It is also called disuse atrophy, especially when you cannot use that muscle much, then it dies because it's lost its nerve supply, hence it cannot be used, so it begins to die. So it's known as disuse atrophy. Since I've used the word atrophy, let's look at some other words with it. Hypertrophy is the exact opposite of atrophy. Atrophy is the muscle dies because the sarcoplasm inside shrinks, blood supply shrinks, all of that. So the size of the muscle just gets real small. Hypertrophy is where there is increase in size of an individual muscle fiber. So it becomes thicker. And that is because of increased sarcoplasm in it, more connective tissue around it, increased blood supply. So all this helps it to increase in size. Hypertrophy. This is normally what happens when you go to the gym and you work out and you do resistance training. Muscles hypertrophy. So for example, if you want to kind of build up your biceps and you're pumping iron, if suppose there were 100 muscle fibers in the biceps and your bicep suddenly begins to look much larger than it was, it's not that the 100 becomes 200. The 100 fibers remain 100, but each fiber increases in size, so the entire muscle then looks larger, okay? So hypertrophy is something that is normal in us, especially when we are pumping iron and, wanting to build up muscle. It may be abnormal when, say, the cardiac muscle becomes very large. It hypertrophies. Usually that will happen when it is trying to push blood against resistance. So that's not very good for us. But otherwise, with skeletal muscle, which I'll talk about, hypertrophy is the normal way to go when you kind of build up and increase the size of the muscle. There is another term which is called hyperplasia, which is increase in number of fibers. Hyperplasia does not happen physiologically in us, in most muscles. So again, to give the same example, when you work out in the gym and build up your muscle, hypertrophy is what's happening, not hyperplasia. So the number of fibers don't increase, it's only the size of every individual fiber which increases. In our body, the only organ which or one of the only organs, for your purposes we'll take it as being the only organ, the only organ where muscle fibers increase both in size and increase in number is the uterus, the pregnant uterus. So when a woman gets pregnant, because the uterus has to house the fetus, she can't do with just increasing the size of her muscle fibers increase in number as well. So the uterus undergoes hypertrophy and hyperplasia so that it can accommodate this growing fetus. When the fetus is born, when the baby is born, the uterus tries to come back to its normal size, but it will never come back to the size it was before because now she's got increased number of muscle fibers. Let's say she had 100 to begin with, maybe she got 150. So they may become smaller in size, but the number still remains more than what she began with. So it will never come back to what's called the pre-gravid state. Okay? So physiologically in our body, for your purposes, the uterus is the one organ which undergoes both hypertrophy and hyperplasia. Otherwise, normally muscle fibers only undergo hypertrophy. Let's look at the three types of muscles that we have. Skeletal muscle, which is structurally striated and functionally voluntary. It's under your control, and we'll see that it has a banding pattern. Smooth muscle, that's why it's called smooth, non-striated, involuntary. Cardiac muscle is striated, it has a banding pattern, but it is involuntary. So skeletal muscle, where do you find skeletal muscle? On the skeleton, all over the body. So in your limbs, on the outside, in the trunk area, in the abdomen, limbs, neck, face, the diaphragm, which separates the thoracic and abdominal cavities, all this is skeletal muscle. Smooth muscle, where do you see smooth muscle? Very good. In the walls of organs, blood vessels, okay? It's in the walls of blood vessels, walls of organs like the stomach, intestines, all of that. Cardiac muscle is seen in the heart and just the beginning of the great blood vessels which arise from the heart. So that's cardiac, but principally we can say the heart. Another example of smooth muscle is a muscle which is seen in your skin, in the dermis of your skin, which is known as the erector pylorem muscle. The erector pylorem muscle is a smooth muscle. This is special smooth muscle. So wherever, again, you get these special names, always pay attention, always highlight that. So erector pylorem is a special type of smooth muscle which is seen in the dermis of your skin. And the name, as it suggests, erector means to erect. Pyli means the hair shaft. So it actually, when it contracts, it makes your hair stand up straight. So when you're scared, for example, you get goosebumps. That's because of contraction of this erector pylorem making the hair stand up, and because of its contraction, it pulls on the other side and raises the little bump on your skin. This erector pylorem muscle is special also because it's only supplied by a part of the nervous system called the sympathetic nervous system. Sympathetic nervous system. And sympathetic nervous system, when we do the nervous system, we'll do it in more detail. But sympathetic nervous system is the one which kicks in when you have a fight-flight reaction, right? So when you're scared, your hair stands up, so then that's the way you can kind of remember this. So here let's look at the structure of muscle. So when we look at a whole muscle, so I've already talked of sarcolemma, the classification, myoglobin. There's another thing that I want you to know, which is called mycium, and you'll see this in your notes. So just to kind of show you what I mean by mycium. When we take muscle, and when you go buy a steak, those of you who do eat steaks, and if you look at a nice juicy steak like this, you would notice that if you haven't noticed it now, next time you go and take a look, you kind of, what you call marbling is because of the fat which is present in between. But also you'll notice that the steak is kind of divided into little groups of muscle inside it, right? Okay? So what happens is that if we take a whole muscle, let's say I'm taking the biceps. If this is the entire biceps and I cut a section through the biceps and this is the whole biceps muscle like this, this is covered on the outside by a connective tissue layer which kind of acts like a little skin for it on the outside. So there is a connective tissue layer right on the outside of the entire muscle. This connective tissue layer is called epimysium. Epi, the way you're going to remember epi, like epidermis, which is on the outside. So epimysium surrounds a muscle on the outside. This muscle then is broken up into actually bundles, which is what I showed you in the steak. A muscle is actually made up of many bundles like this. Bundles are known as fascicles. And each bundle, this epimysium sends in connective tissue around each bundle. So it's really from the epimysium, connective tissue goes around and now it surrounds each bundle. So this is now called perimysium. Peri meaning going around. So epi is around the entire muscle. Perimysium is going around each bundle which is seen in this muscle. Each bundle in turn is actually made up of muscle cells. So each bundle actually is made up of tiny muscle cells. So you can see all these muscle cells actually give rise to the entire muscle. So surrounding each individual muscle cell is another layer of connective tissue which is called endomysium. So this in blue is called endomysium. Okay? Endomysium, which surrounds each individual muscle fiber. Epi, and the way you're going to remember, epi meaning like epidermis, so surrounds the whole muscle. Peri, going around, surrounds each bundle. Endo surrounds each individual muscle fiber. Endo for inside. So if I give you this example as I showed you for the steak, and can I erase this? So remember I drew this steak like this, right? And I said this was broken up into bundles that you see in the steak. So your epimysium would be around this entire muscle here. So this is epimysium. This going around each bundle is perimysium. But in a steak you can't see the individual fibers. You can't see each individual muscle fiber. That's more microscopic. But if you could, then around each individual muscle fiber, this would be called endomysium. Okay? So mysium just is connective tissue, and this is how it's arranged around the muscles. Now let's take a look at this individual muscle fiber. So I'm going to look at this individual muscle fiber, which I told you was also called a muscle cell. So when we look at skeletal muscle, this is skeletal muscle. So this muscle is actually present as long cylinders. Instead of being a short cell like this, it's really a long cylindrical cell this way. And that's because actually when skeletal muscle was being formed, these cells kind of stuck to each other and the cell membrane between them broke off. So now it became one long cylindrical cell. Notice this banding pattern, so that's why we say it is striated. Also, because many cells actually stuck to each other, the nuclei now are all present in this long cylinder. So one single skeletal muscle cell is multinucleated. It has many nuclei. So notice up here, 1, 2, 3 nuclei in this muscle cell. One in this because you can't see the others. The nuclei is present just below the cell membrane or sarcolemma. So the nuclei are what are known as hypolemmal. Hypo means below, lemma means sarcolemma. So the nuclei are hypolemmal, just below the sarcolemma. So here is the cell membrane, just below that you can see the nuclei. So this is typical of skeletal muscle. Let's compare this to another striated muscle, which is cardiac muscle. So this one here is cardiac muscle. Here the cells are short branching cells. By that I mean, if you take a cell, if this is one cell, this is another cell, it might show a little branch like that. This is a third cell, may show a branch like that. Okay? One nucleus in the center, But this also has a banding pattern which you can see like this. So here observe a cardiac muscle. So this is one short cell, this is another short cell, one short cell, here's one short cell, here's a little branch that you can see. So this, and then notice this lines going across. So this is also striated. Single cells which show branching. And where cells meet one another, you can see this line which is present where cells meet one another. That line is called the intercalated disc. So this cardiac muscle shows intercalated discs. Only in cardiac, yes. So this area is where the intercalated disc would be. Okay, only in cardiac muscle, yes. So that's why it's very important to look at these slides carefully. So notice up here you don't see that, but notice intercalated disc only mentioned in cardiac muscle. Smooth muscles, called smooth because it doesn't have any striations. The cells are spindle shaped, so they are long spindle-like cells like this. And the nucleus is in the thickest part of the cell. So they are spindles this way. They really look like fish in a stream almost. Single nucleus, no striations. So that's typical of smooth muscle. So that's muscle tissue. Any other further information is there in your notes, so be sure to kind of read all of it. Next is nervous tissue. In nervous tissue, we have two types of cells. One is the cell which conducts impulses. So these are the conducting cells which we call neurons. And then we have cells which are supporting cells. They don't conduct impulses, but they support the neurons carry out their function. So these are known as neuroglia or neuroglia. So the property of these neurons is obviously conduct impulses, react to stimuli, and that is the irritability property that they have. In some areas of, normally when a neuron goes and reaches another neuron, it sends or it releases something known as a neurotransmitter. So where one neuron meets another neuron, it releases. So if there's one neuron here, it meets another neuron, it releases a neurotransmitter which allows it to send the impulses through. But in certain areas of our body, the nervous tissue, those neurons secrete hormones also. So when we do the nervous system, we'll do that in more detail. But now, for now, this is kind of general basic about the nervous tissue. So let's look at a neuron and look at some features of that. So when you look at a neuron, and I'm going to draw a typical kind of neuron like this. It has a cell body. This is the cell body with the nucleus, nucleolus. It has all of the normal things that every cell has. But in a neuron, it has other things which are known as processes. So this is the cell body, which is also known as the soma. These are processes. The processes are, the small ones are known as dendrites. And all neurons have one long process which is called an axon. This neuron will come down and then it will meet another neuron. So let me draw another neuron up here. It will meet another neuron. Where it meets another neuron, that area where it meets another neuron, there's no cell-to-cell contact. There's a small gap between the two neurons. That area is called a synapse. So where one neuron comes close to another neuron in order to pass impulses across, that region is known as a synapse.
Can you say that again?
Where one neuron comes close to another neuron, there is a little tiny gap present across which the impulse passes, information passes from the neuron above to the neuron below. That gap is called a synapse. And that's where these neurotransmitters are released, and that's what helps to pass the information. That is, there are different types of synapses, but where neurotransmitters are, that's called a chemical synapse, yes. Inside the cell body of a neuron, the rough endoplasmic reticulum is very, very prominent, and it is seen as granules. In the cell body of neurons, the rough endoplasmic reticulum is very prominent, seen as granules which are known as Nissel's granules. Because remember, protein synthesis, neurotransmitters have a lot of protein in them. Rough endoplasmic reticulum is important for protein synthesis and transport. So that's why you see so many Nissel's granules here. Now, neurons are not always the way I've shown over here. Over here, you can see there are multiple processes, right? Many of these dendrites and one axonal process. Such a neuron with many processes is known as a multipolar neuron because it's got numerous poles, multipolar neuron. This is seen in the gray matter of the central nervous system. CNS stands for central nervous system. Sometimes neurons may have only two poles. So you have a cell body, one small dendrite, and one long axon. Such a neuron is called bipolar. It's seen in the sense organs. So can you tell me where you would find sense organs? Pardon? Skin is, let me put it as special sense organs. Very good. Olfactory epithelium in your nose for smell, olfaction, so olfactory epithelium. Then tongue for taste. So on the tongue, yes. I think what you mean is vision. So in the eye, okay, in the retina of the eye. So vision in the retina of the eye. Because your orbit is the entire area. This whole thing is the orbit. Okay. Ears, yes, very good for hearing and also for equilibrium, you know, to maintain balance. So for hearing and equilibrium. So these are the areas where you will find in all these areas, in the olfactory area, in the tongue, in the retina, in the ear, your special sense organs which all have bipolar neurons. And lastly, we have something called pseudo-unipolar. So remember this word pseudo, which means it's not truly unipolar, but it looks like. So pseudo-unipolar is one where the neuron begins, this is the cell body, it has only one process, so then we call it unipolar. But as soon as this one process is given off, it divides into a dendritic and an axonal process, something that resembles a dendrite, something that resembles an axon. So therefore it's called pseudo-unipolar, because not truly unipolar, because it's split immediately into two parts. And this is seen in a part of the nervous system called sensory ganglia. For now you can just remember it as sensory ganglia, because when we do the nervous system we'll talk about it in more detail. Then we have these supporting cells called neuroglia. So here these supporting cells are of different types. So let's look at them. One which looks like a star. This is called an astrocyte. Astro for star. It looks like a star. This is important for providing nutrition to the neuron. So it provides nutrition to the neuron. So it's got many tentacles like this. So it's kind of star shaped. So what it does is one end goes to a blood vessel, so it wraps around the blood vessel. The other one is very close to a neuron. So it kind of takes the nutrition from here and transfers it to the neuron. So it helps with nutrition, the main function. Second is a cell called microglia, which is the phagocytic cell in the central nervous system. This is phagocytic. Astrocyte, microglia, these are present in the central nervous system, which means in the brain and spinal cord. So we see these cells in this area. A third kind of cell is called oligodendrocyte or oligodendroglia, cyte or glia. This lays down myelin in the central nervous system. Myelin is a fatty coat which is present around the axon. So it's like, a wire that your electrical wire, the black coating around it, imagine something like that, so that the axon is not naked. So if this is imagine this is a multipolar neuron like this, and this is the axon. There is a sheath, an insulating sheath which has gaps around it, little gaps like this. This insulation is known as myelin. So in the brain and spinal cord, this myelin is laid down by the oligodendrocyte. Yes.
What does it mean that it stains darkly?
It just stains dark with the stain that you use to stain nervous tissue. The nucleus stains very, very dark. It's very small, microglia, extremely small. So you can't tell in a slide, you can't really tell these cells, and I'll show you the picture. You can't tell these individually from each other, but you can definitely make out the neurons because they're much larger. Yes, all three of these are present in the brain and spinal cord. Yes. Is this fatty sheath which is surrounding the axon? It's like a it's insulating, and it helps to transmit impulses fast, because the impulse kind of jumps from this area which is known, this little space which is present here is known as the node. It's like a node, it's called the node of Ranvier. So myelinated fibers tend to have impulses which jump from that. Imagine it's like an overpass. You can travel much faster instead of going on a straight road like that. So this myelin is like a little overpass.
So the nodes of Ranvier, those are along
Those are the areas where it is bare.
So they're along the axon, though?
They are all of this is along the axon, yes.
And then the
Oligodendrocyte is the one which lays down this myelin. Yeah. In an area called the peripheral nervous system, which is not the brain and spinal cord, in an area called the peripheral nervous system, which is not the brain and spinal cord, this is the nerves, where the nerves are, there this myelin is laid down by another cell which is called the Schwann cell. So here is a difference. So wherever there is a difference like this, you must know. So myelin in the central nervous system is laid down by oligodendrocyte. Myelin in the peripheral nervous system, which is where nerves are present in the periphery. Brain and spinal cord are present in your skull and vertebral canal. The nerves come out to your limbs and your trunk and all this area. There the myelin is laid down by a special cell called the Schwann cell. So it's different. Wherever there is a difference, you must know. Okay? Peripheral nervous system. It's called peripheral nervous system. Peripheral nervous system. So these are the main neuroglia that we see. We have some others called ependymal cells and satellite cells. If it's mentioned in your notes, read about them, okay?
So then the peripheral nervous system is everything else?
Yes, it's everything outside of the brain and spinal cord. And we usually put the nerves as the main component in that. So here, if we take a look, so here are multipolar neurons. So you can see here are the multipolar neurons. The neuroglia look like little dots. You cannot make out the neuroglia on a slide. So up here again, if you look at a slide of the brain, so you can see these multipolar neurons. You can tell they are multipolar because you can see many processes up here, and this is one axonal process. The processes are so many, so they kind of form a little network all around. The neuroglia just look like little dots. You can only see the nuclei, so you cannot tell which neuroglia is which. Okay, so in order to actually be able to see the structure of neuroglia, you really have to do a more detailed and more high power, probably electron microscopy. Okay?